Good morning, students.
Good morning, sir.
Good morning, sir.
So till now, we were talking about the various steps, the tools which are used in the biotechnology. There are some more similar tools or techniques which we will be discussing now. But now we are mainly going to focus on the process of biotechnology, or basically the process of genetic engineering, wherein we are going to discuss various steps. Okay, we are calling it as the steps of recombinant DNA technology, or recombinant DNA technology. In other words, we also call it as genetic engineering. Now recall, we said that we will take some kind of an example, such as you want to take an insulin molecule and then you put it inside a bacteria so that the bacteria can produce large number of insulin molecule for us. This introductory diagram here in this page which you see, it shows a similar type of a mechanism. We see that there is a cell. From this cell, let's say this one, you can say, for example, this is a human cell, suppose. Okay. Then from this human cell, you are isolating a gene. So let's say this is a insulin gene, for example. Then on the other hand, we have a bacteria from where we have isolated a plasmid. This plasmid we are going to use as a vector. What we are doing in the next is, we are taking the insulin and the vector together and binding them together here. As a result, we are getting what we call as a recombinant DNA molecule. This recombinant DNA molecule will now be introduced into so this is a process which is called as recombination. After recombination, we have introduced it into a cell. This cell is a host cell. So this introduction process is known as transformation. And inside the bacteria, we have this recombinant DNA technology, recombinant DNA. In this recombinant DNA, the specific gene of interest from here to here, this is undergoing this process here, which is called the gene expression. In the second chapter of genetics, you have studied, or you will study, this particular process, how a DNA molecule gives you an mRNA molecule. From that, after translation, you get a protein product. So in our example, this is how we got our insulin protein. Now after this, the last step that we are going to discuss is how to do the mass production of this molecule. We are not going to produce it in the laboratory in very small quantity. We will need it in large quantity. So mass production is necessary. Now this is in brief the basic steps that we are talking about, right? But if we go into more deeper understanding, we have to start thinking about, suppose in the first step itself, first thing that you need is how you are going to isolate the DNA from the donor cell. You are not going to get the gene directly, right? You cannot get the gene alone. You have to first take out the DNA. From that DNA, we have learned how we can use certain enzyme with the help of which you can cut down the DNA into certain gene fragments, right? So that we are going to discuss every step now in more details. This is the same thing what we were talking about in stepwise manner. These are the things you are going to understand. So obviously the first step that comes into picture is isolation of DNA molecule. Okay, this is done from what we call as the donor cell. This is done from the donor cell. Okay. In fact, this process we are going to learn in brief in the practical also. Right? Now, if we look at it in more details, the process of DNA isolation basically depends upon what type of host cell you are going to use. Okay. Based upon host cell, you will use different types of enzymes for the separation of the DNA. Right? Now, suppose if your host cell is a plant cell. Now, understand very simple. Where is the DNA present? DNA is present inside the nucleus, and nucleus is present inside the cell. So obviously the first thing that you have to do is break down the cell. Correct? So for that you have to break down the outer boundaries of the cell. Now if we are talking about the plant cell, then this plant cell will contain cell wall. Right? This is not host cell. This is a donor cell. The cell from where you are going to obtain the gene. Now, plant cell you have to break down cell wall. So cell wall is made up of what? We know it is made up of cellulase, hemicellulase, pectin, etc. So for breakdown of the plant cell wall which contains cellulose, you will need an enzyme which is known as cellulase. Correct? It also contains pectin. So you want to break down pectin, you will require an enzyme which is called as pectinase. Correct? So these enzymes will be necessary to break down the plant cell. Then if I talk about a bacterial cell, if you want to take the gene from a bacterial cell, then bacterial cell is surrounded by a cell wall also. So if you want to break down the bacterial cell, we normally use an enzyme which is called as lysozyme. Okay. Similarly, if I say I am trying to get a gene from a fungal cell, fungal cell is made up of cell wall whose composition is chitin. So you will need an enzyme which is known as chitinase. So like that, your different donor cell will have different characteristics based on which you have to decide what type of enzymes will be required to take out the gene from such type of cells. If I talk about animal cell, suppose we are talking about an example of insulin gene. So you have to take that insulin gene from animal cell. So if you want to separate the DNA from animal cell, there are certain types of detergents which are used. Okay. Common, for example, there is one detergent such as we call it as SLS, SDS, etc. Okay. Now, obviously, therefore, first thing we understand here is you require to treat the tissue with this type of enzymes. There are certain steps such as you have to do first of all a process which is called as tissue homogenization. You have to do tissue homogenization. Now based upon the type of tissue, if it is plant tissue or fungal tissue or animal tissue, which has a large number of cells in it. Bacterial cells are unicellular, so you don't need tissue homogenization. There is no question of having a tissue. Mainly in plant and animal cells, tissue homogenization is a process by which you can break the tissues, and when you break down the tissue, you will get the individual cells. Okay, first of all, you will break down the tissue. Then after that, what will you do? You will go for enzyme treatment, right? As discussed previously in our book, they have discussion of only this particular point. That is, what are the various types of enzymes which might be used in case of different type of cells. Now, once you have done the enzyme treatment, you will be getting the cell broken down into its components. So let's say you have taken a vial, and in this vial, in a medium, we normally use here a medium which is called as an extraction medium. This is called as an extraction medium. Extraction medium means it is a medium in which you can extract the DNA, right? So in the extraction medium, you will have here the various cells. These are tissues first, then you have broken them down into cells. Once you have done the enzyme treatment, what will you get? You will not have cells now. You will have all the cell components, right? Because the cells have been broken down. So from these cellular components, you have to separate out the DNA molecule. So very importantly, there are multiple steps of what we call as centrifugation carried out. Okay? Centrifugation is carried out. When you do the centrifugation, this helps in separation of the DNA. This will help you in separation of DNA molecule. Correct? Now, at this point, you have obtained the various types of DNA molecule. But do not forget, these DNA molecule are often also associated with various proteins as well as RNA. So you will also need to carry out a treatment with protease as well as RNAs. As a result, all the unnecessary proteins or RNA which might be present along with the DNA, that will get separated, that will get degraded in fact. So you can get from here, by doing all these steps, you will get what is called as the purified DNA. Right? Let's say you are doing it, you are trying to find out animal cell, from animal cell, human cell, let us say, from there you are trying to take out the insulin gene. In that case, you may start with, let's say, blood cells. Again, if you want to do blood cell, then the tissue homogenization step will not be required. Okay, so this is the first step, wherein we have isolated the purified DNA from the cell. Now this purified DNA, we have to break it down with the help of restriction endonuclease enzyme. Correct? So that will become therefore the first or second step. So first step was isolation of DNA molecule. Right? Now, second step will be cutting of DNA molecule. We have already discussed in details about the restriction endonuclease enzyme and how to obtain a particular gene fragment from there. So you will cut down the DNA with the specific restriction endonuclease enzyme. Now please keep this in mind. When you are doing this, ideally you are supposed to be aware, or you are supposed to know about the sequence of your gene of interest. Okay, and the sequence which is present on the either sides of your gene of interest. Right? For example, let us say you have a DNA molecule, and this is from here to here you have the gene of interest. This is your gene of interest which you want to cut down. Now to cut out this gene of interest, what do you require? You need to cut the DNA at this point and at this point, right? Now if you want to cut at these 2 points, then you need an enzyme which can identify the sequences present on the both side of your gene of interest, means at this region and at this region, correct? Which means you must know the sequence so that you can decide which enzyme to use, because you have on one hand the names of different enzymes and their recognition sequence. That database will be available, right? The enzymes and their recognition sequence database is available. Now once you know what is the sequence here, according to that you can identify which enzyme will have this particular recognition sequence, and accordingly you can decide which restriction endonuclease enzyme you have to use, correct? So on one hand you will cut out the specific gene of interest from the DNA which you have just now isolated from the donor cell, and at the same time, in the previous step when we spoke about isolation of DNA from the donor cell, you also need to carry out separately plasmid isolation. Plasmid is also a DNA. So this plasmid isolation also you have to do, because you will need a plasmid, no? So this plasmid isolation from some bacterial cell, you have to do both. Now the plasmid is also a DNA, so a similar process you have to carry out. Now when you study in more details in higher classes, you will learn that there are certain differences in terms of genomic DNA isolation and plasmid DNA isolation, right? So that details we will not discuss here, because that is mainly your difference in the practical aspect, actual practical aspect. So you have now in your hand the DNA isolated from the donor cell and plasmid isolated from the bacteria. So when we say cutting of DNA, you are doing it for two processes. Number one, you will cut the DNA to obtain the specific gene of interest or fragment of interest. At the same time, number two, you will use that plasmid DNA and you will use the same enzyme to cut that plasmid DNA or the vector DNA so that you can open up the vector DNA, right? Now, we have also studied in the previous classes that once you have cut off the DNA to obtain various fragments or genes, various fragments, what you have to do to get this or isolate the specific gene from all the fragments which you are getting. For example, if you look at this diagram, this was one whole DNA molecule. When you have cut, it got cut into multiple location. So when you cut it at multiple location, you can see there are so many fragments formed. Now let us say this is actually your gene of interest out of all these fragments. So you need a technique by which you can isolate this particular gene of interest, correct? So after this process, you are going to do what? Agarro gel electrophoresis. Recall the tools we have studied already. Now remember, if you forget the tools which we have studied earlier, then here in the process you will not be able to understand. So why we are using the gel electrophoresis step here? Because with the help of that gel electrophoresis step, we can separate out the specific gene of interest from the entire fragments, all the fragments that we have obtained by cutting the DNA molecule. So two steps we have discussed. One is isolation of DNA, other is cutting of the DNA to obtain the specific gene of interest. If anyone has any question here right now in these two steps, you can ask me now before we proceed further.
So tissue homogenization is for like increase the efficiency of the process now.
No, you see, you cannot get. Let us say, suppose you will in fact do this process in the class also. We will make you people isolate the DNA from some plant tissue. When you want to isolate the DNA from plant tissue, you need to have the cells, but you cannot get isolated individual cells, right? You will be taking some tissue such as very commonly we take banana tissue, one of the most commonly taken tissue. Or we will take some very young leaves, small young leaves we will take. So there you do not get individual cells, you will get tissues. So that tissue you have to break down first. In animals, in human beings, we normally take blood because that is easy to take. If you want to take some other animal tissues, we also sometimes do an experiment by obtaining the DNA from other animals, like we take goat animal from the goat tissue. So we normally start the experiment by taking the goat liver. So when you have a piece of goat liver in your hand, the first thing you have to do is what? Break down that tissue, right? So when you break it down, you will get individual cells first, and then the second step will be to break down the cells. So directly, if you do a enzyme treatment to the tissues, then all the cells of the tissues may not break down, right? That's why tissue homogenization is required. Okay, so let us now talk about another new thing which you are learning for the first time, but like we know, it has become a very common word in today's world. That is PCR. All of us have been hearing about since last 2 years one new word, that is RT-PCR, right? If you do not do RT-PCR, you cannot get into an airplane, you cannot go into certain particular place, you need to submit the RT-PCR report. Now, RT-PCR is nothing but a special type of PCR. So let us today first understand what is the meaning of PCR. Its expanded form is polymerase chain reaction. There is a process where a chain reaction keeps on happening with the help of an enzyme known as polymerase enzyme. And that's why we call this process as polymerase chain reaction. To speak the truth, it is again a very, very revolutionary process which was discovered by the scientist known as Carey Mullis. But the process was so obvious kind of process, okay, that when Carey Mullis got Nobel Prize for discovery of this process, he himself was surprised. He never thought that he might get Nobel Prize for discovering this process, which is kind of such a simple thing actually. Okay, now understand what happens in this polymerase chain reaction. The point is, sometimes your gene fragments with which you want to work. Okay? Any kind of work. Let's say best will be to understand, let us say we want to do some forensic study. And in that forensic study, you have obtained some sample from where you have isolated some genes, some DNA. Now, there might be a situation wherein the amount of genes available or the amount of gene fragments available is very, very less. It is very less. So, you will need a process to amplify the amount of genes available or make some copies of that gene. Understand? So, polymerase chain reaction is basically a process wherein we can make copies of a particular gene fragment. How it is done, let us understand that. Now, look at this diagram first. This explains the entire process, how it is done. The first step, there are three basic steps. The first step you have to do is known as denaturation. What do you mean by DNA denaturation? DNA denaturation means separation of the two strands of DNA. So you start with this DNA molecule. This is a double-stranded DNA molecule. Okay. Here what you are doing, you can do the denaturation of DNA by two processes, remember, in general. One is if you provide alkali treatment. If you want to do denaturation, either you can provide alkali treatment because DNA is acidic in nature. So if you do alkali treatment, then also you can break down DNA, or you can do heat treatment. Right? Now in this case, we are doing a heat treatment. So our temperature, which is more than 90 degrees centigrade. Here it is mentioned 95 degrees centigrade. A temperature greater than 90 degrees centigrade will lead to, look at the next diagram, what is happening, the two strands getting separated. Okay, here both the strands now got separated. Now what is the ultimate aim is, based on one particular strand, considering one of the strand as a template strand, a new strand will be synthesized to get the two DNA molecule. For which the second step that we have to carry out is called as primer annealing. This is normally called as, the step is called as annealing. Annealing, basically the word means kind of joining. Now here you have to add in the reaction something called as a RNA primer. Now what are these primers? These RNA primers are very short sequences of RNA. Very short sequences of RNA. Okay, temperature is little reduced. From 90 degree, it is brought down to around 50 to 70 degrees centigrade. Okay. Now these primers, these are found ready-made in the market. When you purchase these primers, in those primers, the temperature to be maintained will be written. This is called as an annealing temperature. Means the temperature at which the primer will get attached to the DNA molecule. So you can see here the primers have got attached to the two ends of DNA, individual DNA strands. Right? Then comes the third stage, which is called as extension. Right? These are the three steps: denaturation, annealing, and extension. In the extension, what happens? You will again change the temperature and provide here in the mixture a DNA polymerase enzyme. Understanding? This is a DNA polymerase enzyme. Now, what is the function of DNA polymerase usually? It helps in polymerization of deoxyribonucleotides. Because what is DNA? DNA is basically nothing but a polymer of these deoxyribonucleotides. Now we are talking about synthesizing a new strand of DNA, which means you have to polymerize certain deoxyribonucleotides. So it's not only you are not only adding the polymerase enzyme, you are also adding in the mixture various DNTPs. What are DNTPs? That is nucleotides basically. DNTPs are nothing but the free nucleotides. Okay. These are free nucleotides you are adding to the reaction. Okay, and with that you are adding the polymerase enzyme. So that polymerase enzyme will actually go and bind at this location and at this location in both the strands, and then it will, okay, it is shown in this diagram. At this location it will bind, at this location it will bind, and then it will start adding the nucleotides in a specific direction, as you can see here in this direction and here in this direction. As a result of this, what do you get? You get two strands, completed two strands, right? And thus you get two molecules. You started with one molecule, you get here two molecules of DNA. Fine. Now this particular process will be repeated in a cyclic manner. Okay, one cycle of polymerase chain reaction is over, and this process will be repeated again and again. That's why this arrow is shown. That's why we call it as a cycle. When this is repeated again, from two molecules you will get four molecules. So we understood that PCR is basically a process by which DNA amplification can be done, and during this process we can make multiple copies of DNA or a gene fragment. Right. Now what we see here is there will be mainly three steps: denaturation, annealing, and extension. As far the words are concerned, denaturation is the process by which the two strands of DNA can be separated. Annealing is a process which means joining something. Extension is extension. It's I mean you extend something. Okay. So during denaturation step, with the help of a heat treatment, we expose the DNA to a high temperature by which the two strands of the DNA is separated into individual strands. Then we use a short sequence of RNA molecule which is called as a RNA primer, and at a specific temperature this primer is attached to the template DNA. We call these as template DNA. Okay, the original DNA strand based on which the new strand of DNA is synthesized. That original strand is called as a template DNA or template strand. So to the template strand, we have added here the RNA primer, at two different ends. Then comes the main process where we are adding free nucleotides, which will be acting as monomers for the new strands. And we are using a polymerase enzyme, which helps in polymerization, means joining of the individual nucleotides. Then, with the help of the primer, the enzyme polymerase will start adding nucleotides one by one in a particular direction, thereby giving you a whole new strand of DNA. Clear? Now, the most important thing about the polymerase chain reaction, PCR, which comes in the exam regularly, is about the enzyme polymerase. Now, try to understand, if you look at the temperature here, you can see the temperature is all very high temperature. Polymerization we are carrying out at 72 degrees centigrade. So, it is logical to understand that at such a high temperature, an enzyme cannot work, because enzyme is a protein, and you have studied in class 11 that enzymes are very, very temperature specific. At high temperature, it undergoes denaturation. Correct? It will not work. So, this was a big challenge to discover or to find out way how, by maintaining high temperature, the polymerization can be done. Now, why we cannot bring down the temperature? Because if we reduce the temperature, what will happen is the renaturation process. From double-stranded to single-stranded is denaturation. If I say from single-stranded to double-stranded, return, that process is called as renaturation. So, if we did the denaturation by maintaining a high temperature, if you bring down the temperature, it will show a process called as renaturation. It will get renatured. So, you cannot bring down the temperature. You cannot allow this process of renaturation to happen. But you also have to do polymerization. So, the challenge is, if you bring down temperature, then polymerization will happen. But before polymerization can happen, the two individual strands will undergo renaturation. And if you keep the temperature high, then two strands will remain separated, but the polymerization process will not happen. Okay. So what happened is, after a lot of search, Carey Mullis, who was considered as the discoverer of this particular technique, has found that there is a bacteria known as Thermus aquaticus. Okay, these are the bacteria which are found in hot springs, where the temperature can be very high as around 90 to 100 degrees centigrade. So he thought that if this bacteria is naturally found in such a high temperature, it means that the various enzymatic processes in these bacteria will also happen successfully at high temperature. So what he did is, he isolated the DNA polymerase from this bacteria, and that DNA polymerase is known as Taq polymerase. So the polymerase enzyme which we are using here is actually a Taq DNA polymerase, where Taq stands for Thermus aquaticus. Okay, this is the most important point of this topic. What is the significance of Taq polymerase? Very important. So Taq polymerase is obtained from the bacteria Thermus aquaticus, which is found in hot springs. As a result, this polymerase enzyme can naturally work or carry out polymerization at a very high temperature. Clear? So this is the process of polymerase chain reaction. If we look further, this is how the process keeps on happening. First cycle, second cycle, 2 cycles are shown. You look at this. This gives you another understanding of this process. Okay. What do we see? You start from one template DNA. This is one molecule. The red colored area is shown as the wanted gene. After one cycle, you get 2 copies. After 3 cycles, you get 8 copies, and so on. Okay, when you continue to do these cycles, okay, multiple cycle, you see just 35 cycles are done, only 35 cycle. There is a way to calculate. After the first cycle, you get 2 to the power 2, 4 copies. Okay, after second cycle, you get 2 to the power 3, 8 copies. Right, and so on. When you keep on continuing, you get in a very short span of time. Each cycle, I can say, each cycle usually takes approximately, you can say it approximately takes around 20, 30, 40, around 1 hour, approximately. Around 1 hour, right? So approximately. So within a short span of time, if you run it for 24 hours, okay, means in 24 hours you will get 24 cycles. So a huge exponential amplification, a huge number of copies can be obtained in a short span of time. And this particular phenomena wherein you can get huge number of copies, okay, large number of copies of DNA in a very short period of time, actually. This is responsible for making various diagnostic processes fast. Okay. It has made fast diagnosis of various problems, commonly like cancers and all. Earlier diagnosis, which used to take like almost 1 week, 10 days, like that, now those diagnostics can be done in just few hours. You have seen in RT-PCR only, when you did, you can actually get the result within an hour when you, after you give the sample. So fast you can get the result. So this fast diagnosis is one of the biggest application of PCR technique. Okay. So this is the instrument. Now it is a fully automated process, wherein this instrument is actually called as a thermocycler. Thermocycler means the timing can be, the temperature can be cycled. So you can see it is associated with a computer, wherein you can do the programming in such a way that you will give instructions such as, for example, this is just an example, 95 degrees centigrade for 5 minutes, then bring it down to, let's say, 70 degrees centigrade for 10 minutes, then 75 degrees centigrade for around 15 minutes, for example. Okay, so total, This is half an hour, 30 minutes, if you see, in which one cycle is complete. You give the instruction in such a way that after this 15 minutes, 75 degrees centigrade, it will go back again to 95 degrees centigrade 5 minutes. And thus this will become a cycle of temperature. So that's why we call it as a thermocycler machine. Okay. You can open the lid from here. Here you have a lid. When you open the lid, inside you have a facility to put some small vials like this. In these vials, you will add a mixture. That mixture will contain your template DNA, right? Plus the primer, plus DNA polymerase enzyme, plus the free nucleotides. All of these will be present here. So you mix everything at once itself, okay, and you put it there in the machine, and you set the timing. And what will you see? You will see this cycle keep on happening. So this entire cycle, you can also set that this will continue for, let's say, 30 cycles, for example, right? So how much time how much ever time is required for that time, just switch it on, make sure that power does not go in between, and then you go home. Don't worry, it will continue to happen. You can even say, you can even add to the program that after 30 cycles are complete, then bring it down to 4 degrees centigrade and keep it like that for, say, 10 hours. Okay. So you can design your experiment like before you go home in the evening from the laboratory, you put these things in place, switch on the cycler, make sure there is a power backup at all the time, and then you go home. Next morning you come, you will see that the process has been completed, and the final result, the large number of molecules which are obtained, they have been kept at temperature was 4 degrees centigrade. What is that? Refrigeration temperature. So it means it has been refrigerated after the process was complete. So that's how in actual this process is carried out. Understand? Like in your class when I was telling that you can always go to some institutes and see these things. You can also actually do an experiment on what is called as do a project on what is called as instrumentation. Only instrumentation, nothing else. Means you are going to learn different types of instruments which are used. This is only one instrument which I told you. Okay, like this, there are so many different instruments used for different purposes. When you study biotechnology and certain other concepts, you will learn about those instruments. Okay. So that is all. If we go back, we started with how the DNA is isolated, how the cutting of DNA take place, and if necessary, please remember, this is not a compulsory process. Polymerase chain reaction is an optional process. If necessary means if at all the DNA fragments are less in number, then you might go for amplification to make multiple copies, right? Then in that case, this will be the third step. Okay. So we spoke about polymerase chain reaction in details, very important topic. I can say almost 90% chance you will get a question on PCR. Then after that, the next step will be, we have isolated vector, we have isolated the gene of interest. Next step, join them. Okay, so this step will be to join that gene of interest with that of the vector. Now this is a diagram which is there from your book, okay, where they have shown that how basically two molecules are joined together. I have discussed this earlier also, right? Basically, what in this diagram, what they are trying to show. Now, this diagram might create a confusion. In this diagram, you might get confused because we said that restriction endonuclease enzyme, it is an endonuclease enzyme, right? It is an endonuclease enzyme. Endonuclease enzyme means it will cut the DNA inside somewhere. But in these diagrams, they have shown two different molecules. One is white, one is black. And they have shown the cut at one end, not in the center somewhere. So do not get confused. This diagram is not to make you understand the function of enzyme. This is basically to make you understand that when you use the same enzyme to cut two different DNA molecules, you will get this type of complementary sequences in the sticky ends. And when you bring them together, they will spontaneously bind because of the complementarity between the bases. Okay? That is why this diagram is shown to make you understand that if an enzyme is used to cut two different DNA molecule, same enzyme when you will use, then there will be complementary sequences formed, and those complementary sequences will easily help in binding of the two molecules. That is the understanding we get from this diagram. You can better concentrate on the second diagram here. Here what is shown, please observe carefully. What is done? You have a foreign DNA. This is your DNA isolated, right? From here, we have done ECO-R1 restriction endonuclease treatment is done here. As a result of which you got fragments of DNA. From this, you can do electrophoresis. When you do electrophoresis, you will be able to isolate your specific gene of interest. So this gene of interest is this one in blue color. On the other hand, you have a plasmid vector, which is also cut open using ECO-R1 enzyme only. As a result, you can see there is an opening at this region. Okay. If you observe carefully, since both the molecules are cut using the same enzyme, therefore here also you get TTAA sticky end, and here also you will get AATT sticky end, which is complementary. So that's why they are binding here spontaneously. Similarly, AATT here sticky end, AATT here sticky end, so they complementarily bind together here. Is it understood, everyone? If anyone has a question or doubt regarding how the DNA fragment binds to the vector, please clear it here. So final molecule that you will get, this is your what we call as recombinant DNA molecule. Many people call it as a plasmid chimera, or sometimes people call it as a chimeric DNA. So this is one step where the vector ligation means joining, is carried out. Right? Please recall, at this moment, you should not forget that this process is not happening manually. If you look at the practical lab activity, then what is happening? You have here a vial which contain in the buffer various plasmid with that enzyme. its plasmid has been treated. And here you have another vial, okay, where you have the buffer with your gene of interest. What is done? The two vial buffers are mixed together, and here you have one vial where you have in this buffer the mixture of open plasmid plus the gene of interest, where you are expecting that this process is happening. What do you call this process? This process is called as recombination. But recall, you cannot see this actually happening. You are only expecting that recombination has successfully happened. So do you recall how will you ensure that this recombination has successfully happened? In one step, what was that process known as? What will be the name of the process which is carried out to ensure that the recombination has successfully happened? We call it as selection. Recall, we studied about something called as selectable marker. So this is called as selection process. But you cannot carry out selection process right at this step. Why? Because selection is done for the particular bacterial cell. We are growing that bacterial cell in a particular medium containing particular antibiotic. Right? Now here the bacterial cell has not yet come into picture. We have only spoken about recombinant DNA molecule. After this, immediately the next step which you are going to do is transformation. So this is the step of what is called as transformation. In the transformation, what will happen? You have the vial containing the supposedly recombinant DNA molecule. I am saying supposedly because we do not know for sure. And then you have another vial containing here host cell. Here also recall, these are supposed to be competent host cell, because remember we have made the host cell competent, then only it can take up the recombinant DNA. Without that it will not take up. So that's why those tools were discussed separately. Now you mix these content here and expect the transformation to happen. After this, what will you do? You will be doing what? Selection. Okay. You will do the selection to identify those bacteria or those host cells, okay, which contain the transformant and recombinant DNA. Right? So this is where the basic process completes actually. Fine? You have in your hand now those bacteria after selection which is containing your gene of interest, the bacteria which contain the plasmid with your gene of interest. These colonies of these bacteria, you can either allow them to grow, okay, they will be allowed to grow in the growth medium, and you put them in growth medium, they will divide and they will give you clones. So you will get many copies of that gene of interest, and you can maintain. You can maintain it, right? Try to understand here. I will tell you one last thing for today. Now, after doing all this, what is, are you able to see my whiteboard? Can anyone give me a feedback? There
Yes, sir.
is some issue in the whiteboard. Yeah, it has come. Just hold on. Yes. So I have now with me the colonies of transformant and recombinant bacteria. Now it depends upon what is my experiment aim, what I want to do. I can either go for making what is normally called as genomic library, okay, or in our book this is not mentioned. Genomic library is not there. In our book we are talking about obtaining foreign gene product. Okay, so this is what is there in our book, but we have a separate topic called as human genome project. So to understand human genome project, we have to understand what do you mean by genomic library and how it is maintained, right? So very simple, genomic library discussion details we will do later. I mean the human genome project we will discuss later, but understand what is basically done is for maintaining a genomic library, what is the meaning of this genomic library? I will give you a very simple example. Let us say human beings. Human being contain different chromosomes. Let us say you take one specific human chromosome, one particular human chromosome, some chromosome, you take X chromosome, say for example, right? You take X chromosome. So this is your X chromosome. With the help of restriction endonuclease enzyme, you cut it down and you get many pieces. So you get multiple fragments of the DNA here, right? Now, it is not a question of gene of interest. Here there is nothing called as gene of interest. All fragments are of interest only. These are all fragments which we require, right? Now what we will do after this, we will add them to different vectors, right? Let us say for example 1 2 3 4 5 6, 1 2 3 4 5 6. So 6 different vectors you are going to add it. Then you will transform them. Transform them means you will put into different hosts. How many different hosts you will get? 6 hosts. Correct? So from 6 hosts, what will you get? After successful transformation, after successful recombination, transformation, selection, etc., what will you get? You will get 6 different colonies of host, right, containing 6 different fragments of X chromosome. Okay, so when you maintain these different colonies of host, you have to maintain it in a library. Maintain means you have to ensure that they survive till forever, you can say. Because whenever you, it's like, for example, you want to get some information. You want to get some information on Shakespeare, for example. So where will you go? You will go to that library on this earth which contain 100% all the creations of Shakespeare. Yes or no? So same thing, you want to get some information about X chromosome, some gene, some fragment. You want to work on that. Okay, what will you do? You will come to this genomic library. This is a library which is containing the genome. You will come to this genomic library which contain all the fragments of X chromosome. As per requirement, you can obtain the particular fragment and carry out your research work on it. So this is the concept of what we call as genomic library. When we will learn about human genome project or any other organism genome project, then the first thing to do or first thing to make is a genomic library, where the main aim is to maintain the fragments of the entire DNA or to maintain all the genes either wholly or in the form of fragments, okay, in a particular host cell. This is about the genomic library. In the next class, we will be talking about how to obtain the foreign gene product, which is there in our book. Genomic library making is not there in our book. So that was all for today's class. Next class, we'll continue about the obtaining foreign gene product.